All right, let's dive deep. I mean, really deep. We're going to journey into the bustling, microscopic city that is a single human cell. And we're on the hunt for a tiny, often overlooked hero that is absolutely essential for our survival, the peroxisome.
So let me just start with a question for you. Right now, this very second, your cells are creating toxic chemicals. It's just a normal byproduct of being alive. So, how do they manage that? How do they deal with this constant internal poison without just, self-destructing?
Well, the answer lies with that unsung hero I mentioned, the peroxisome. The best way to think about it? It's the cell's dedicated hazardous waste and recycling specialist, working 24/7 in the background to keep everything running smoothly.
So what is it exactly? Well, a peroxisome is a small organelle, basically a little bubble with a single membrane around it. They were first spotted back in 1965, and their entire reason for existing, their one main job, is to take all those dangerous substances floating around and make them totally harmless.
Now, this is where it gets really interesting. You've probably heard of the cell's powerhouse, right? The mitochondrion. Well, look at how different the peroxisome is. While mitochondria are all about generating energy, peroxisomes are purely focused on cleanup. And get this, unlike mitochondria, they have zero DNA of their own. That means every single tool, every protein they need to do their job has to be shipped in from the outside.
So, let's get into the nitty-gritty of that cleanup job. And I'm not exaggerating when I say that these little guys handle some seriously hazardous materials for us day in and day out.
So who's the #1 villain in this whole story? It's a molecule you've probably heard of, actually. Hydrogen peroxide. Inside our cells, it's being made all the time as part of normal metabolism. And if you just let it float around, this stuff can cause a huge amount of damage. It's super reactive.
And the peroxisome solution is just, well, it's elegant. It's a perfect two-step process. First, some of its own enzymes create that very hydrogen peroxide as a byproduct of their work. But, and here's the genius part, the peroxisome is also packed with another enzyme called catalase. Catalase immediately grabs that dangerous molecule and poof, turns it into harmless water and oxygen. Problem solved, just like that.
But it's not a one-trick pony. The peroxisome is a real specialist with a few key jobs. Besides handling hydrogen peroxide, it's also the main place in the cell for breaking down a specific kind of fat, these things called very long-chain fatty acids. If those build up, they become toxic. So yeah, this little organelle is a true metabolic workhorse.
So this all begs the question, right? If these things are so important, where do they even come from? For a very long time, this was a genuine scientific mystery.
For decades, scientists were basically split into two camps. One group was convinced that peroxisomes were kind of like bacteria. They'd get bigger and then just split in two to make a new one. But the other camp argued, no, they're built totally from scratch, budding off from this other big structure in the cell called the endoplasmic reticulum, or the ER for short.
So who is right? Well, in a really cool twist, it turns out they both were. Modern science has shown us that it's a unified process. The cell can build brand new peroxisomes from the ER, and existing ones can grow and divide. It just uses whichever strategy it needs at the time. Okay, so that brings us to the next big puzzle. How does the cell actually build one of these things? How does it make sure a new peroxisome gets all the right tools it needs to do its dangerous job?
And here's the kicker we talked about earlier. Peroxisomes can't make any of their own proteins. They don't have the DNA blueprints, and they don't have the little factories, the ribosomes, to build anything. This means every single enzyme, every structural piece, has to be made somewhere else in the cell and then shipped over.
I mean, think about that for a second. That's a huge logistical challenge. The inside of a cell is an insanely crowded place. How on earth do you make sure the thousands of different proteins being made all get to the exact right place? How does that catalase enzyme know to go to the peroxisome and not wander into the nucleus by mistake?
Well, the cell has this incredible system. It's basically a super efficient postal service. Each protein that's supposed to go to the peroxisome has one of two special shipping labels, called PTS1 or PTS2, built right into its code. Then, specific mail carrier proteins, PEX5 and PEX7, recognize those labels, grab the protein, and make sure it gets delivered to the right doorstep. It's an absolutely amazing system of precision.
Now, I know this all sounds super microscopic, maybe a little abstract, but here is why it all matters so much. Because sometimes, that assembly line, that postal service, it breaks. And when that happens, the consequences for a person's health are devastating.
A tragic, real-world example of this is a condition called Zellweger syndrome. It's a rare genetic disorder where, because of a mutation, the body simply can't build working peroxisomes. That whole intricate import and assembly line we just talked about, it fails.
And without those working peroxisomes, remember those very long-chain fatty acids? They can't be broken down anymore. They start to build up to toxic levels, especially in the brain and nervous system, and that leads to severe, heartbreaking health problems. It's just a stark reminder that our overall well-being completely depends on these tiny, unsung heroes doing their job perfectly every single second of our lives.
The peroxisome really is the perfect example of a critical piece of machinery working quietly in the background, keeping us safe. It just makes you wonder, doesn't it? What other microscopic heroes are operating inside of us right now, performing these incredible, life-saving tasks that we are only just beginning to truly understand?